Okay, so for this video, I'm going to try and go over mitosis and meiosis, and let's just get right away started into some of the differences here. So each cell, they're going to start with just regular amounts of chromosomes, so 2N, and that means a maternal and a paternal chromosome. Remember, because you have a pair, one from mom, one from dad. Each one is also going to go through S phase, so that is times 2 DNA. And so what that's going to do is add another one here. See, that kind of like crosses. Another one there. So now you have sister chromatids. These are copies of each other, of maternal and paternal. Same thing over here. You have a small cross over there. So now you have essentially, I don't like to call it 4N, but in my head, that's what I usually call it. So in this case over here, everything's going to line up on metaphase. And the maternal copies are going to line up here, and the paternal copies are going to line up here. This is mitosis. And they're going to split this way and split this way. And what you end up with is another 2N cell and another one. So in this one, remember that it's just going to split up evenly, and you're still going to end up with the same amount of chromosomes, and nothing's going to change in this one. So for meiosis, it's a little different. Since you have these pairs and you need to do this again, you're actually going to end up with 2 cells that still have 2N. But in this case, these are the sister chromatids, and up here happened all that, like crossing over, independent assortment, or actually I think assortment comes later. So then you have this one, so these are each 2N. And then they're gonna split one more time, and now you only have one chromosome, and you can't control whether it's paternal or maternal. And these are all 1N, and there's 4 of them. So those are some of those key differences between the two. And then up here, you also have crossing over between these two, like right here, like these little segments where you can flip paternal sections of the chromosome with the maternal sections too. So crossing over, and then they also independently assort at this stage here. I think in meiosis too, let me check and make sure that's when that happens. M phase, meiosis 1, separated. Sorry, I just want to be sure. So I think, yeah, so this says that crossing over and independent assortment, that all occurs right here in meiosis 1. So you get crossing over, and we'll call that independent assortment. And that all happens right here at this stage. So remember, this S phase is important because the DNA is doubled, and you get these little sister chromatids. These ones split up evenly, so maternal and paternal still go into the exact same copies of the cell. This one, the actual pairs line up, they split, then they have to split again. And that's important because you have to do that, that section one more time. Okay. Another thing to remember is that when we were looking at those onion cells, remember that the cell cycle, and despite the fact that we're studying mitosis, mitosis itself is not a very big section of the cell cycle. You have G1 where the cell's growing, G0 where you can just rest. The cell like just won't do anything, it'll wait. S phase is pretty small, this is when the DNA doubles. And then there's a really long phase called G2 where there's lots of like checkpoints, you know, things like that, that the cell makes sure it can divide again. And then here is where DNA doubles. So definitely, this is kind of tough to do all on a whiteboard, especially the mitosis, meiosis stuff. Definitely watch animations and videos, find resources. That PDF I sent you guys will show you a lot of differences between meiosis and mitosis. So here's another example crossing over. Say here are your paternal copies. And this is crossing over in meiosis. And remember, this doesn't happen on different chromosomes. This happens on the same information chromosome. So, but it's paternal one, the maternal one. And it's when you switch sections, and there's like cellular machinery that causes like a switch and a break right here, for example. And then what you would get is, start there, and now that's a paternal section of DNA, and now that's a maternal section. And that's crossing over. And then remember, when these split up one more time, because you see how there's 4 right here, these will all split up, and you'll get 1, you see this direct strand, you'll get 1 pure maternal, but then you'll also get this one with a little bit of paternal on it, and you'll get this large paternal with a little bit of maternal on it. So hopefully that makes sense, and how crossing over can lead to variation. And here this also visualizes independent assortment. You can't control which chromosome is going to end up being in that haploid cell. Okay.